Hello everyone, welcome to today's class. Today we are going to see the steps for genetic engineering, or we'll discuss how we need to isolate genetic material. First we will discuss steps for recombinant DNA technology. Steps for recombinant DNA technology. So here first we'll be discussing about isolation of genetic material, right? Now these are the steps. First we will isolate genetic material DNA from the living organism, then fragmentation of DNA by restriction endonuclease, separation and isolation of desired DNA, which we will do it with the help of gel electrophoresis, then amplification of gene of interest, ligations with vector, transfer into host culture of host cell in nutrient media. That means what is the criteria here? See, if this is a cell and this is the DNA, we want a gene. Suppose, let me first mark this gene here. Suppose this is a gene of our interest. So what we do first, how we will take out this gene? Our motive is to take out this DNA. So what we will do first, we will first isolate the DNA, right? This entire DNA we will isolate. Yes or no? Let me change the color with green only so that it's become easier for you guys to understand. So just a minute. Yes, so this is the DNA, right? And suppose this is our gene of interest. Now I have isolated this DNA. Now this DNA is the total DNA which is present in the cell. Now what we have to do, we have to undergo the fragmentation. We have to cleave this DNA into small fragments like this. We have to cut this DNA into small fragments so that we can get our desired gene. And this fragmentation is mainly done by an enzyme. We call this enzyme restriction endonuclease. They will cut this DNA into small fragments. Now what we do, see here, there are many DNAs now. There are so many DNAs present here, right? So how will we identify our gene of interest? Out of all this DNA, we need to isolate our gene of interest. How we will do it? We'll do it with the help of a process known as gel electrophoresis. That means we will separate the DNA in the presence of electric field, right? After gel electrophoresis, what we will get? We will get our desired DNA. This is what is our desired DNA. Now this is only the one piece of DNA, one piece of gene. Now we need to increase its number so that if any error happens, we no need to again repeat the steps. So what we will do, we'll just run this in a PCR machine. PCR, which it is mentioned as amplification of gene of interest. It is done by PCR. So here we will apply PCR. Now after PCR, what we will get? We'll get many copies of the genes, many copies of the desired genes. That depends on how many cycles we run in the PCR machine, right? Now what we do, this gene of interest, now we will transfer it to a vector. What is the vector here? Vector is a kind of vehicles. We take normally plasmids, that is from bacterial cell. If this is a chromosomal DNA, this is a plasmid. Now what we do, we take this plasmid and this will be considered as vector. So this is the plasmid. Now again what we do, we cut the plasmid with restriction endonuclease and you will be getting this kind of structure. Now what we do, we ligate or we paste our desired gene in this vector. How this pasting is done? This is done with the help of an enzyme known as DNA ligase, right? And after that, we will transfer this inside the host cell, again back into the host cell. Now what we do, we culture this, we culture this bacteria in a nutrient media so that the cell increases its number and transfer its genetic material to other cells. Once it increases its number, what will happen? Our gene will be amplified again and we'll get more number of genes, right? So now then again, once the bacteria start growing or start culturing in the nutrient media, it will start expressing, after some time it will start expressing the protein encoded by the gene, and after that we extract the protein, right? So this is the steps we have written here. First is what? This is isolation of DNA. So first is the step here, say isolation of DNA, right? This is fragmentation of DNA by restriction endonuclease. Then third is what? Gel electrophoresis. right? Then fourth is the PCR machine. Yes, this is the gene of interest I have shown. So this is fourth is PCR machine. Then ligation with vector, right? Then this transfer into the host, right? And then we will grow it in a nutrient medium in a bioreactor. And this is what we call sixth. Now this bacteria will start growing, increases its number. Say from 1 you will get many bacterial cell here. By feeding and by taking nutrients, they will increase its number and start expressing the protein. After some time we will recover the proteins, which is term test 8, which we can call as 8, and this would be 7. This would be what? 7. Clear now? So this is the process of gel electrophoresis. Now this is the steps here. Now we will explain, we'll see each in detail. So here, actually here we have, again, we will see how we will isolate the DNA. That is, the first step is isolation of DNA, right? So for isolation of DNA, we'll see this in detail, how we will isolate the DNA. If this is a cell, now this is cell membrane and this is genetic material, right? So this is cell wall and this is cell membrane, right? Now what we have to do is, first we need to digest the cell wall, right? For digestion of cell wall, we'll be using different enzymes with respect to the cell. If the cell is bacterial cell, we'll be using the enzyme lysozymes. If the cell is a plant cell, we'll be using cellulase. And if the cell is fungal cell, we'll be using chitinase. Why? Because bacterial cell is made up, cell wall is made up of peptidoglycan. So to digest peptidoglycan, we use lysozymes. And plant cell wall is made up of cellulose. So to digest cellulose, we'll be using cellulase. Again, fungal cell is made up of chitin. So to digest chitin, we'll be using chitinase, right? So these are the enzymes we'll use to digest the cell wall of the bacteria, which is here number 1, right? After cell wall is digested, we'll come across the number 2, that is cell membrane. So cell membrane is made up of lipids and proteins. Cell membrane is made up of proteins and lipids. So to digest the cell membrane, what we'll be using? We'll be using protease and lipase, right? And here cell organelles are also present. So here, if any proteins or lipid structures are there, it will digest all the structures made up of lipids and proteins. Next, we will be having RNA here. So we don't want RNA. So we again will be using ribonuclease, also known as RNAs. We'll be using RNAs to digest RNA. To digest RNA, right? So here our interest is what? What is our interest? Our interest is the DNA. Is DNA. So we will not be using DNA. So DNAs will not be used. Will not be used, right? So this is the first steps we will do for isolation of DNA as per the cell. If it is a bacterial cell, we will be using lysozymes. If it is a plant cell, we will be using cellulase. If it is a fungal cell, we will be using chitinase. Then we will use protease, lipase, RNAs to digest the rest of the materials. Then we will be having our DNA, right? Now this DNA, if this is a beaker or a test tube, now here you will see some transparent thread-like structures, right, all over the tube. Now we need to precipitate it, make them in one particular area like this. How we have precipitated? How we have precipitated? We have precipitated this with the help of chill ethanol. ये पीने वाला नहीं. This is not drinkable one. Okay, this you cannot drink. So this is what we call spooling. Spooling is basically treatment with chilled ethanol to collect the DNA. Now we will get this DNA. Right? So this is what we call spooling. Now once we get our DNA, what we have to do now, we have to fragment the DNA using restriction endonuclease. So all restriction endonuclease has got definite recognition site, and we will see which recognition sites it will work, and we will cut it. Suppose this is 5 base pair, this is 6 base pair, this is 2 base pair, right? So here we will apply restriction endonuclease to cut the DNA into small fragments. Now, once we get these small fragments, here we have what? Here we have a process called gel electrophoresis. Because we have now, we need to separate. These are suppose fragments of DNA. Fragments. Suppose these are DNA fragments. Now we need to isolate our gene of interest. Our gene of interest. How will you get our gene of interest? Through a process known as gel electrophoresis. Agarose gel electrophoresis. Right? So what is agarose gel electrophoresis? Here we use a gel. Say this is a gel here. And this gel is made up of agarose. This gel is made up of agarose. And this is extracted from the seaweed or red algae named as Gracilaria and Gelidium. Gracilaria and Gelidium. Right? And this completely agarose gel will put a comb here. This kind of comb are present, and we'll put a comb here, and this comb will create wells here. Right? What we will do now, DNA is what? DNA is negatively charged. What is the charge of DNA? Negative. So when we apply electric field, this DNA will start moving to a positively charged anode. Right? And this movement is based on size and molecular weight. Smaller the fragment, larger is the movement, or faster is the movement, right? Larger is the segment, slower is the movement. Clear? So if you understand gel electrophoresis, gel comes from the word agarose gel. Agarose gel. And electrophoresis, electro means electric field, and phoresis means movement. So that means movement in gel under the application of electric field. We call it gel electrophoresis. Gel means agarose gel we used for this process. Electro means electric field. Phoresis means movement. That means movement of a charged particles in the presence of electric field in the gel agarose. Right? Now here what we are using? Here we are using DNA. So we call it DNA gel electrophoresis. Now DNA is negatively charged. DNA is negatively charged. So on application of electric field, it will start moving towards the anode that is positively charged. And what is the medium we have used? The movement will be inside the agarose gel, right? Because they have agarose have a small pores, and through this pores DNA will start moving. Right? And this movement of DNA is based on size and molecular weight. Very important. This movement of DNA is based on size and molecular weight. Suppose if I go back to the slide, this 2 base pair is my gene of interest. Suppose this is my gene of interest. Now what it will do, when I place all the wells, all the samples here, what will happen? These samples will start moving. Now this is start moving. This is quite, you see, this DNA, this fragment of DNA is coming fast, is moving fast. That means this DNA is our gene of interest, because rest all base pair, 6 base pair, 5 base pair is all cleaved by restriction endonuclease, right? Restriction endonuclease, we recognize which restriction endonuclease will apply and we'll get this much base pairs. Now 2 base pairs are our gene of interest, so this being a smaller in size, so it will move faster. Out of all these, see, it is moving faster, so this is our gene of interest. Right? So this is how DNA gel electrophoresis works. That means in the applications of electric field, the charged particle, charged DNA will start moving to the oppositively charged anode, and here what will happen? The movement is basically through a gel, we call it agarose, which is obtained from the red algae Gracilaria and Gelidium. Right? Now here, this DNA being transparent, we cannot see it with our naked eye. So here we use a stain, we use a dye, which is known as ETBR. What is that? This means ethidium bromide. Right? When this ethidium, we stain this DNA with ethidium bromide and we expose this DNA to a UV transilluminator. Under that, you will see orange color DNA bands. Orange color DNA bands, you can see that, right? And after, suppose if this is a agarose gel here and these are wells, these are our DNA, right? So this is our DNA interest. What we will do, we'll cut this particular gel, and this process of extraction of DNA pieces from agarose gel, we call it elution. right? So what is elution? Extraction of DNA piece from agar gel, we call it elution. Next, now we have our gene of interest. We got, now this is our gene of interest, we extract it, yes or no? Now we need to amplify it. We need to produce more copies of this isolated DNA. So what we will do for more copies? We will use a machine known as polymerase chain reaction, or PCR, right? So what is the function of PCR? It makes billions of copies of DNA gene, right? And this process was discovered by Carey Mullis, right? And here, since this process takes place in 3 steps, what is the 3 steps? D, A, E. This is a code you can remember. D for denaturation, A for annealing, and E for extraction. And E for extraction, right? So we call it DAE, denaturation, annealing, and extraction, right? So denaturation, the temperature is quite high, 94 degrees centigrade. Denaturation, we will explain what it is. We'll explain it. Annealing, the temperature, here you can see we expose this DNA to a different temperature, right? And we'll see this in our coming slide. So here, as the temperature is high, you can say 55 to 60 degrees centigrade. So as temperature is high, the enzyme which we will be using here must be thermostable. Must be what? Thermostable. But that means the enzyme needs to be stable at high temperature. And for that, we are using a specific enzyme which we call Tech polymerase, This is a thermostable enzyme, and for PCR we are using this enzyme. Why it is called as Tech? Because this Tech is from the word, from the bacteria. T comes from thermus, AQ comes from aquaticus, because we have isolated this enzyme from bacterium Thermus aquaticus, right? So it is known as Tech polymerase, which is used for PCR machine, right? And along with Tech polymerase, what are the other things we need for PCR? We'll be needing a primer. Primer is nothing but a short oligonucleotide sequence, right? And we'll be needing nucleotides. That is what we call DNTPs, deoxyribonucleotide triphosphate. Basically, these are DATP, DGTP, DCTP, and DTTP, all nitrogenous bases, right? Now here the 3 step denaturation. If you see, denaturation is what? This is our double-stranded DNA. Yes or no? This is our double-stranded DNA. When denaturation means what? These 2 strands opens up. When we expose this DNA to a temperature of 94 degrees centigrade, these DNA strands get separated. That means the hydrogen bonds break. When we expose this double-stranded DNA to a 94 degrees centigrade, what will happen? It leads to the breaking of hydrogen bond. Hydrogen bond breaks. As a result, what you get? You get 2 separate strands, right? Now what we will do? Again, once the DNA strands are separated, we will once the DNA strands are separated, that means denaturation process is done. Our motive in denaturation is to break this hydrogen bond so that the 2 strands get separated, right? Then what we do, next step is your annealing. Annealing means what? We reduce the temperature to 55 degree to 65 degrees centigrade. And here in this temperature we add primer. What are primer? It is a short oligonucleotide sequence which go and bind to the 3' hydroxy N, right? So this is primer. It will go and bind to the 3' hydroxy N. Fine. And this process here, primer is forming a base pair at 3' hydroxy N. So this process we call it annealing. Clear? And here temperature is 55 to 65 degrees centigrade. Correct? Next comes your extension. In case of extension, what is happening? We will be adding here DNTPs, that is nucleotide. That is what? Nucleotide will be adding, will provide here DNTPs. What DNTPs will do? The Tech polymerase, bacterial enzyme Tech polymerase, it will take DNTPs and keep on adding with the primer. DNTPs, Tech polymerase will pick up DNA and keep on adding in the DNA strand. So from now, what we'll get? We'll get 2 strands. We'll get how many strands? 2 strands. Let me show you here. 3 prime to 5 prime, and let me say 5 prime to 3 prime. How many strands will be getting? 2 strands. One is like this, 5 prime, 3 prime, again 3 prime, 5 prime. So from you see, after one cycle, how many you got? 2 strands. So here the number of copies of DNA you got, number of copies of DNA, how much you get, that is in the form of exponential form, 2 to the power n, and here n stands for number of cycles. Say if 10 cycles we have done, if we have continued 10 cycles, then number of copies of DNA will be 2 to the power 10. Clear? So this is what is the PCR, polymerase chain reaction. 3 steps you have to remember: denaturation, annealing, extension. In denaturation, what we do? We just expose the DNA to a very high temperature, that is 94 degrees centigrade, so that hydrogen bond breaks and we get individual strands. Then we go for a next step, that is annealing, where we expose the DNA strands to a temperature 55 to 65. What it will do? A primer, that is short oligonucleotide sequence, will go and form base pairs with 3' N. Right? So what it will do? 3' N, it will start adding, it will start forming base pairs with the it will start pairing up with 3' hydroxy N. Right? Then comes your extension. Now your extension means what? It is primer will extend the strand by forming a complementary base pairs with the existing base pairs. Here if it is A, T, G, C, so here in case you will come T, A, C, G, like that. And who will help in this process? An enzyme called Tech polymerase, which is thermostable enzyme, and we call it Tech polymerase because it is obtained or isolated from bacteria Thermus aquaticus. What it will do? It will pick up the DNTP complementary to the DNA strand, and it will extend the strand. So ultimately after a cycle, what we'll get? We'll get 2 copies of DNA. Right? So this is the old strand, newly synthesized, old strand. Right? So the number of copies of DNA that PCR produce is exponential, and we can find out with the help of a formula, 2 to the power n, and here n stands for number of cycles. Clear? That's it. Now we have many copies of our gene of interest. What we have to do? Now we have to ligate this gene to a vector, which we can call this vehicle. So normally what vector we use? We use plasmids. Right? So here this is a plasmid we have taken, and this is our recombinant DNA. See, this gene of interest we have inserted into a plasmid, and how it is attached to this plasmid? Through an enzyme we call the DNA ligase. Right? It is done with the help of DNA ligase. DNA ligase will add this gene of interest to the plasmid, right? And this plasmid will put in a new bacterial cell, new cell. Now we'll put in a new cell. So after some time, what you will see? You will see that this new bacterial cell will be having a DNA along with recombinant plasmid. Right? And from where we have taken this plasmid? Say this plasmid, this is another DNA, and this plasmid we have taken out and we cleave it with the help of restriction endonuclease. This is the other part. So here we have attached it with the help of our gene of interest. We have attached our gene of interest. So here you get this, and how it is done? With the help of DNA ligase. Now this is a new bacterial cell, new cell after insertion, new cell for insertion of recombinant DNA. Now this we can call as rDNA, because recombinant DNA, because it contains 2 different DNA, right? We call it recombinant. Now this particular cell we will grow in lab. What we do? This grow in lab, right? How you grow this in the lab? Yes. So here one thing, this transfer into host, no, this process, this we have inserted. Now how this recombinant DNA ligation of gene of interest with vector, these steps basically focus how this part is added to the plasmid through DNA ligase. Now I have shown you, this I have inserted into a new bacterial cell. This I have shown you that I have inserted in a new bacterial cell. How it is inserted, that we will study here. Transfer into host. How we insert this DNA into the host cell. So host has to be made competent. Competent means ability to take up DNA. Ability to take up DNA. We call it competent. We have to make the host cell competent, right? DNA is hydrophilic. Hydrophilic means what? Water loving. And plasma membrane is what? What is the plasma membrane? Plasma membrane is made up of what? Proteins and lipids. Yes. The plasma membrane is hydrophilic. Plasma membrane is made up of lipid and protein. So we basically what we do, we make any cell. If it will not take DNA until and unless, suppose this is a recombinant plasmid here. This is a recombinant plasmid here. Now what it will do, this is a recombinant plasmid. A cell will never take this until and unless we treat the cell. We treat cell with calcium. What is calcium here? See here, treatment with divalent calcium cation. Normally we use calcium. Once we treat this cell with calcium, what it will do, this calcium will increase the permeability of the cell. What it will do, it will increase the permeability of the cell. Got? Now the treatment, according to the cells, we do different treatment. If the cell is bacterial cell, we use some different process. If the cell is animal cell, we use different process. And if the cell is plant cell, we use different process. Right? Now, the most common, let's see, bacterial cell. If we are inserting the DNA to a bacterial cell, what will happen? If we are inserting this into bacterial shell, we'll give a what we will do, we'll give heat shocks. How we will give heat shocks? First we will treat the cell with calcium. First, this is a bacterial cell. First we will treat the cell with calcium. Then we will put the cell in ice. Then we'll put the cell in ice, right? Then we'll give a temperature of 42 degrees centigrade to the cell, and again we keep it in the ice, right? So this is what we call heat shock. This process we call the heat shock. Why we call it heat shock? First we treat it with calcium, important. Then we place it in ice, very low temperature. Again we give a high temperature 42. Again we give a low temperature. So continuous drastic change of temperature. We call it what? Heat shock for bacterial cells. Now if we want to insert this into animal cell, what we will do? If we want to insert this into animal cell, we'll be using a process known as micro injection. This is bacterial cell, right? For animal cell, we'll be using a process known as micro injection. What we do, we'll insert the DNA directly into the nucleus of animal cell through a micropipette. If this is an animal cell and this is the nucleus, right, and here already DNA is present, so we'll take a micropipette and inject it with the help in the nucleus, new DNA, right? So that is what we call micro injection. Clear? Next, plant cell. Plant cell, if we insert it in a plant cell, we'll be using another process for plant cell. What we call that? We call that gene gun. Gene gun means what? Plant cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. If this is gold, this is a gold, it is coated with DNA. Now we will insert this gold, or you can say tungsten with DNA to plant cell. This is a plant cell. See, this is the nucleus of the plant cell. Now what we do, we put this inside the plant cell we bombard this with high velocity, and this will start moving inside the cell. So that is how we transfer the recombinant DNA into the host cells. Fine. Next, how we have to select the recombinants from non-recombinants. Selection of recombinants from non-recombinants using selectable marker. How we do that, last class we have seen. I have shown you, right? So if a gene is inserted, we'll see that gene is functional or not. How we see, I already have made a video with that. You can check that, right? Then culturing of host cells into nutrient media. Now we grow these cells inside the nutrient media, right? And say if you want to see how let me explain you this, because selection of recombinants from non-recombinant, how we will decide whether these are recombinant or non-recombinant. How? Suppose, let me give you an example. Say take a plasmid. Suppose if I give this example here, let me take a plasmid PBr322. Right? Now in this plasmid, say this is a plasmid here. Right? Now this part is a gene. We call it TET-R. That means this gene is resistant to tetracycline. And this is a gene, it is resistant to ampicillin resistant antibiotic. It shows ampicillin resistant and it shows TET-R. That means what? Tetracycline resistance. Right? Now, in this tetracycline resistance gene, there is another site which is a recognition site for enzyme BAMH1. This is a recognition site of enzyme BAMH1. Right? Now, what I will do, I will ligate a gene here. Suppose I want to ligate the gene at BAMH1. Suppose this is a foreign gene here. I want to place it here in this BAMH1 site. This ligate the new gene at BAMH1 site. Right? Now what will happen? When I inserted this new gene to a BAM1 site, what will happen? This site got disturbed, yes or no? And as a result of insertion of this gene, what will happen? The entire TET-R gene is affected, because normally TET-R gene, TET-R means the gene which is resistance for antibiotic. Normally it had BAM1 site and it was function normally. But once I inserted the new gene here, the BAM-I site got disturbed, as a result of which TET-R got disturbed. So what will happen? TET-R or tetracycline resistant gene is destroyed. Is destroyed. Yes or no? It is destroyed. Now, if we grow this bacteria, in presence of antibiotic tetracycline, if we grow this bacteria in the presence of tetracycline, what it will do? It will not grow. It will not grow because the tetracycline resistant gene is destroyed. Right? So here what is happening? TET-R gene is destroyed. Why it is destroyed? Because we ligate the new gene at BAMH1 site. So here TET-R is destroyed. So because of destruction of TET-R, tetracycline, TET-R resistant gene is destroyed. So it will not grow in the presence of tetracycline. It will not grow in the presence of tetracycline. So now if you grow, now this cell, after insertion of new gene, if you grow this cell in the presence of ampicillin, do you think cell will grow or not? Cell will grow. Why? Because ampicillin resistant gene is still functional, because we have not inserted any gene here. So it will grow in presence of ampicillin. But in case of tetracycline, if we grow this cell in the presence of antibiotic tetracycline, do you think this will grow? It will not grow, because the site is being disturbed. Now what we will do? We will now isolate the recombinants from the non-recombinants. Clear? This is what we call selection. This is how we can select recombinants from the non-recombinants. Clear? Because recombinants will not survive in the presence of tetracycline. Clear? Same, we can also use another factor you can use, which we call PUC, Plasmid, from University of California. Now let me explain this. Suppose this is another plasmid. Let me write this PUC. Now here let me explain you this. It has a gene which shows resistance for ampicillin. right? And now it has a gene. This gene is for enzyme beta galactosidase. Beta galactosidase, right? So this gene is what? Beta galactosidase. Now we have inserted beta galactosidase is an enzyme which break lactose into glucose and galactose, right? Now here suppose we have inserted a new gene here. This is a new DNA, new gene. We will put it inside this new gene inserted. New gene inserted, new gene inserted at the beta galactosidase site. New gene is inserted at the beta galactosidase site. Now, what will happen? What will happen? Now, once new gene is inserted, do you think beta galactosidase is functional? Now, beta galactosidase is not functional. Beta galactosidase is not functional because of insertion of new gene. Because of insertion of new gene, right? Now how will you identify this? How will you identify this? Let me draw this here. Say this is a plasmid with beta galactosidase and this is ampicillin. This is again a plasmid after modification. This is beta galactosidase, this is ampicillin R, and this is beta galactosidase. Now what will I do here? Let me introduce the new gene here. New gene, right? Now what will happen? Initially, let me divide this. Initially what will happen? In presence of ampicillin, antibiotic, plus signs for present. In presence of ampicillin antibiotic, what will happen? Yes, what will happen? The bacteria will grow. Yes, because gene is active, bacteria will grow. Now what will happen for beta galactosidase? We will be using a X-gal. What is X-gal? It is a chromogenic substance. Chromogenic means it induces color. Chromogenic substance, that means it gives color. When beta galactosidase gene is active, that means it is breaking lactose into glucose and galactose. So if this is active, how we know that the gene is active? If we grow this bacteria in the presence of egg cell, if the beta galactosidase is active, it will give blue color colony. Blue color bacterial colony. Blue color bacterial colony. But here, what happened? This gene is active, so it is giving blue color colony. But let me see the other case here. Here again I am using ampicillin. Again I am using ampicillin antibiotic. So now you see here ampicillin part is all same. No changes here. No changes done. No changes done. So in the presence of ampicillin, the bacteria will grow. Yes. But in case of X-gal we use for detection of beta galactosidase. But in this we have inserted new gene. That means changes were made. Yes or no? So what will happen? Because of insertion of new gene, this beta galactosidase is inactive. It is not active. So what will happen if we grow this type of bacterial cell in X-gal, which is a chromogenic substance which used to give color if gene is active, chromogenic substance which used to give blue color if beta galactosidase is active. Now here gene is inactive. As gene is inactive, so it will give white color. White color. Why? As Gene is inactive. So it gives what? White color, right? And this is how we differentiate this recombinant cells from the non-recombinant cells, right? This is one way. And in the plate you will be getting, this is a supposed plate, you will be getting culture media, you will be getting both blue and white colonies. This is blue color colonies, and which are non-recombinants. Now recombinants will be having what? White color colony, because their gene is disturbed. Beta galactosidase gene is disturbed. So this is what we call blue white screening. Yes. So this is the image you see. This is a Lec-5 gene. We call that is beta galactosidase. Now this is functional. So what it will do, as this is functional, it will X-gal, it will give a blue color, right? Now this is Lec-5, and here what we have done, we have inserted a gene here. This is a new gene. So what will happen? Gene becomes non-functional. So what will happen? X-gal will not give blue color. Blue color, instead it will give what? White color, right? This is what is the image. Clear? So now we have seen how this recombinant DNA is being, how this is how we will identify the recombinants from the non-recombinants, right? Now we will proceed for culturing of the cell into nutrient media. Culture cells in nutrient media. What we do basically, see here, for growing cells in nutrient media, we use two types. We use a bioreactor. What we call, we use a device, we call it bioreactor. We call it bioreactor, right? Or also known as fermenter. Also known as what? Fermenter. Clear? What is this? This is a, this looks like this, and here some basic optimum conditions are maintained, right? What are the optimum conditions we maintain here? We maintain optimum conditions. What are the optimum conditions we maintain here? We maintain temperature, pH, then oxygen, vitamins, yes, then substrate. Substrate means the content, right? Salt, etc. These are maintained in an optimum condition inside this bioreactor, right? And apart from this, we have how it is controlled. For temperature, we have temperature controller. Temperature controller. For pH, we have pH controller, foam controller, right? So this is how. Now how we added culture media. Based on addition of culture media, it is of two types. That means fermenter or bioreactor, you can make it two types. What are the two types? See here. One we can call as what? One we can call as batch fermentation. Batch fermentation. And another we call it continuous. Right? What is batch fermentation? What happened in batch fermentation? Suppose this is a fermenter. We add raw material or media or nutrients one time. We add nutrients only one time, right? And we open it at the end. Open at the end. That is what we call it batch fermentation. So here we can say here, see, This is not open in between the process. One first time we will add nutrients, and once the process is going on, we will now extract the product at the end for extraction of product. So as we call it closed fermenter, or we call it closed system, right? But in case of continuous fermenter, what is done in case of closed? Here this is a fermenter. What we do, we add raw material, used, drain out, used, again we add raw material, keep on adding raw materials. Yes, we keep on adding raw materials. Why we keep on adding raw materials? To increase the yield, of the product, right? Now at last we will extract. That is what we call downstream processing, extraction of product, right? So here you can see we have multiple times we had added media, multiple times we have added nutrient, multiple times we have added nutrients, multiple times nutrients are added to increase the yield of the product. So this is what we call open system. Now we open it, we closed it, again we open it, we closed it, right? So this is frequently we open it in between the process, so it can be called as open system. Like the one it is called as closed system, it is called as open system. Clear? That is what is the growth. Now next we will extraction of desired product. How will you extract the desired product? Whatever product is there, extraction of desired product. Extraction of the desired product means what? Whatever, for what reason we have done this, we will extract this product. So this extraction of desired product, we call it downstream processing. We call it what? Downstream processing, right? So here what we will do, we will separate and purify the product. Separation and purification is performed. Separation and purification of the product, right? We do separation and purification of the product, right? And sometimes we also add preservatives, right? Preservatives, we add preservatives in case of foods, packet food also you can see, right? Then quality control, we will check the quality of the product. That is what we call quality control, right? Then we go for clinical trial and marketing. Clinical trial and then marketing of the product. This is what? Marketing of the product, right? Clear? So this is what is the entire process. With this, we came to the end of the steps of recombinant DNA technology. Thank you so much for watching the session. Any doubt, please type in the comment section. Thank you.